So now what we're going to talk about is topic 8, and this is how Creutzfeldt-Jakob disease, or similar mad cow disease, affects the brain. And so during this discussion, we're going to be covering the following. Explain how cells use information in DNA to produce proteins. Identify the components of double-stranded DNA. Explain how hydrogen bonds contribute to the structure of DNA. Use a template strand of nucleotides to create a complementary strand. Explain the roles of DNA, RNA, and the protein and gene function. Describe the participants and events of transcription. Explain how RNA is processed after transcription in eukaryotic cells. Describe the participants and events in translation. Use the genetic code to translate a nucleic acid sequence into protein, and then diagram and explain the relationship between codons and amino acids, and explain why regulating gene expression is adaptive. And in the process of this, we are going to look at a disease and a disorder that affects the brain that has been either reported on previously or there is some historical relevance to it in the past 20 years. And so the very first thing that you want to do here is watch this video, and you can find it in the link below, on can you catch Alzheimer's disease, and then it'll start talking about Creutzfeldt-Jakob's disease and the brain. And you can look below and you can see what some of the very similar what does some of the symptoms look like. You can find this in video 2, video 3, and how quick it is. We'll talk about this disease itself, kind of what it looks like and how it works. And why should we care? It is a disease that can hide itself for years. There's evidence that it can hide itself for up to 50 plus years and then end up taking someone's life in relatively short time, anywhere from 2 months to a year. And sometimes it's really hard for people or doctors to determine that's what you have, because it's something in some parts of the country is a little bit more relevant, or not the country, but the world, than it is essentially for people maybe that live in Texas. Now, with that being said, if you're an avid traveler, especially if you had family that traveled in the 90s and went to England, this would absolutely be something that they potentially could worry about if they ate beef at that time, right? Because there was a big outbreak of mad cow disease that affects cows, and then you potentially can be infected by it by eating infected meat, potentially. And this shows you what the implications of this disease is like. The picture in A is showing you what a normal brain looks like, and B is showing you one that is affected by this disorder. So there's a large amount of neuronal damage and decay that happens. So in order for us to understand this disease, we got to understand kind of what proteins are. And so when we're talking about what proteins are, we want to always start with, well, what is the informational unit that tells us how to make our proteins? And that is DNA. So this is a molecule, and if you remember, it is a type of nucleic acid. And we learned from this at the beginning of the semester when we talked about biomolecules. Every single cell in our body uses DNA to basically store our genetic information. It is basically the recipe or directions on how to make us. And basically, we need DNA to produce proteins. And if you remember, proteins are important because those are the mover and shakers in our system. Now, a review, DNA is stored in the nucleus, and I call it almost like it's agoraphobic. And if you know what agoraphobia is, it's people that are scared to leave their home, right? So DNA doesn't leave its home because if it does, it can be destroyed. So it likes to stay in the cells unless there's a process that basically allows it to move around. And we talk about that, or you talk about that in mitosis or meiosis. And you know, in the nucleus, it's stored as chromatin. And if you remember, N for normal. So when we looked at chromatin in the DNA, when we just looked at a cell, and this is a cell, right? And this is the nucleus, which holds it. You kind of remember it looked like it was all just these squiggly lines. And so that's what chromatin looks like. Now we're going to condense it down and into chromosomes during meiosis and mitosis to allow them to move around. Now, if you can see, it is a double-stranded helix. You can see genes are very small sections on that DNA. That DNA, to make sure that it can get into or be stored in that nucleus, is wrapped around proteins. These proteins are called histones, and they make these little circles called nucleosomes. And then that condenses farther into chromosomes that you can see here. Now, we didn't know everything that we know about protein or DNA in the past. So you can watch this quick video below for the history of DNA and the discovery of DNA in the link below. Now, we know the structure of DNA. DNA is considered a double helix. And that structure of DNA in 1955, it was determined to look this very way. And it was figured out through the studying of chemistry and using what's known as X-ray diffraction. So basically, this is the X-ray diffraction pattern that gave us the structure of DNA. And basically, we know DNA is this like double helix. So it's not looking at it like this. Basically, the eye is here, and it's looking down almost like a barrel. That's what we're seeing here. And you're seeing basically different atoms of this DNA coming up in this X-ray diffraction pattern. Now, that X-ray diffraction pattern was generated by a woman by the name of Rosalind Franklin. And I would like you to watch this video to understand the history behind the structure or the discovery of this structure, because it's actually pretty controversial and an interesting story. And she, at the time, didn't get the attention that she deserved for the work that she did. Now, DNA is composed of what we know now are, and this is a review again, nucleotides. So a nucleotide incorporates a sugar, a phosphate, and a base. Those are the three parts. Now the base is interchangeable. This stays the same, and it's called a deoxyribose because right here there's no oxygen. Now this base can change. It can be adenine, guanine, cytosine, or thymine. So this kind of gives you an idea. So you can see the A's and the G's have these kind of double circles, and then the C's and the T's have these single circles. And what makes them different, if you know there is a phosphate and a sugar on that backbone. That's this part, right? And the part that's changing out is this. And so you can see that here. Now, these little dots here, if you remember, that is a hydrogen bond. And if you remember, hydrogen bonds are strong enough to keep these together, but weak enough that they can be unzipped. And so it becomes unzipped when we do DNA replication, and it will also become unzipped when we go through a process called transcription, which we'll talk about later in this discussion. And the way that these nucleotides form these bonds is what gives it this double helix. Now, these strands are considered anti-parallel to each other, which means if you look, this right here is one of the nucleotides, and on the end, there is a phosphate. And then if you go on down over here, what's going to be on the end of here is this structure, this part of the molecule right here, which is basically one of your hydrogens is going to be the last atom that's there. Now, with the anti-parallel, it talks about it being a 5' to 3'. And that just means, if we're looking at this, that the carbon, that is the 5th carbon, is the last one, and they count it from here. So this is each one of these dots, if you remember, is a carbon. So this is a 1, 2, 3, 4, and there's a 5 right here. So because of this phosphate here, this is a 5', and then you don't even have to know what the end is. That's going to be a 3'. And when they say 5', it's just saying that's the 5th carbon from where, on the ring, from where the nitrogen base is. And this is just saying it's the 3rd carbon from where the nitrogen base is. And it's anti-parallel because it's going to be the opposite on each side. So if this is 5' to 3', then what we can say is that other side that's here is 5', and as we go up here, it is 3'. So these are opposite. And this, the way that the strands are aligned parallel to each other, and they're oriented in this opposite direction, allows it to bind and look the way it is. And the way that the bases line up with each other is what's known as complementary base pairing. And so you always have A and T, N, C, and G that base pair with each other. And I think C and G look a lot alike. And I always kind of think there's a couple things that I remember these, but A and T, I think alcohol and tobacco always go together because I know a lot of my friends, when they drink, they smoke. So that's kind of how I remember those two things together. And you can see, look here, C is with G, G is with C, T is with A is with T. So if you see thymine in something, you have DNA. So a DNA sequence or a gene is a small piece of DNA on your whole genome. A DNA sequence is the order of the nucleotides. Because they have this complementary base pairing they've been able to figure out, essentially you can sequence a new DNA strand by just aligning to that what's complementary to each other. Now, with the idea that A base pairs with T and C and G base pair together, you can figure out how much is in each one. For instance, if I go through here and I have G, C, T, and A, because I have complementary base pairing, I can figure out the percentage of what's there. For instance, if I know there are 10% of the sequence is C, and that's what they can give you, then you can figure out, well, because G and C base pair each other, then 10%, if there are 10% C's in there, there has to be 10% G's. And these add up to be 20%. Well, we know 100 minus 20 is 80%. So then you just divide that by 2, and you realize that you have, in the other two things, 40% and 40%. So because you know that this complementary base pair happens, you can project the percentages of G, C, T, and A if you have one value, and then you can predict what the DNA sequence is going to be from the template strand that you use, or the strand that you're using to create a new sequence with. So what you can do now is, on your handout that you have, you can go through and practice getting your complementary base pair to figure out what those new sequences will be. Now, DNA packs tightly into chromosomes when we go to move them around or make new cells. So when this does this, basically it's compacting chromosomes so that you can copy and put that brand new set of DNA, which is called the entire genetic material in the cell, is called the genome, into a new cell. When they do this, the DNA is wound up really tightly and into discrete packages. Basically, if DNA were to be completely unwound and it didn't form in these tightly packed, if you see this right here, right? Basically, the proteins are histones, and you can see here there are 1, 2, 3, 4 of those, and then it is rotated a couple times around that, and those things are called nucleosomes. So if it did not compact this way, then it would be way too big to fit inside cells. I mean, there's enough DNA in each cell to go to the moon and back from here a few times. So there is a lot of it in there. So it does come packed in this way, or package it in this way, to allow it all to fit. So if you would really like to know what the structure of DNA is, please look below. Now, you do not need to know for any of the exams for this class things like what the area here, let me go, here or here is called, or those really technical terms. But I'd like you to kind of understand the structure of what DNA really looks like. Now, if you remember, and this isn't going to be your handout because I'd like to kind of go back to one of the talks that we had before about what are karyotypes. And the reason why we talk about this really quick is I want you to also understand when we're looking at the genome, right? When they're compacting into, if you remember, those small things called chromosomes, basically we have 46 chromosomes in each one of our human somatic cells, which are diploid. Those are 2N. Let's review, right? So we get half our chromosomes from our mom, half are from our dad, right? Which means we get 23 from mom, 23 from dad, until we have 23 pairs, which makes 46 chromosomes. The first 1 through 22 are autosomes. The 23rd is the sex chromosome that basically determines if you have the biological sex of a female or biological sex of a male. And so these, you can see it starts with 1 up here. The 23rd is here, and they're showing you this is what female looks like. And that's a sign for me, female. This is a sign for male. And you can see in female you have 2 X's. In male you have an X and a Y. So I wanted to just hit on this again because when we're talking about DNA and chromosomes, we're talking about in an individual a full set of chromosomes. And on these chromosomes are going to be genes that give us the directions on how to make proteins. So we need these genes or these chromosomes to separate correctly so we don't have any dysfunction. And we saw in one of the previous talks, if you have an extra chromosome, what can happen. But when the cells separate, we are getting all of our genome, our full gene sequence into each cell. And so we look at what are known as homologous chromosomes, and this is, when we're talking about it, we get 1 from mom and 1 from dad. And on these homologous chromosomes, they're not identical, so the actual sequence is not going to be exactly the same. But they're going to have the same types of genes for the same traits, or they have what is considered homologous pairs of genes for the same traits. The genes are arranged in the same order. So what we mean, like this area right here, may be a gene for eye color. And so you can see here these chromosomes, same length and everything else. Here are sister chromatids, remember, divide, because this one is the original, could be, as you know. When you look at DNA replication, you'll realize this is semi-conservative, so you get one from, you get one side that is going to be the original, and then the other that's going to be the daughter. So you have one, and it duplicates for both of these when we want to make a new cell. But if you notice, different forms of the same genes, the genes, those are called alleles. And alleles can be either dominant or recessive. So dominant is determined by the big letter or uppercase, and that is what's seen. So if you know someone who's dominant, right? If you put them in a group of people, you're going to hear them, you're going to see them. But if you have other people that are kind of quiet, that dominant person is going to be the one that kind of overshouts everybody else. Recessive means something that's only seen if there's other recessive genes there. So this is going to be like a quiet person. The only time you're going to hear a quiet person is if there's other quiet people around. Now they're denoted by lowercase or small letters. So if you look at these are dominant. This is dominant. This one is an example of a recessive. So as we're looking at this you would see dominant. This you would see dominant. This over here, that's when you would see the recessive. Now, if you got one from mom and one from dad, your genes would look like big B, little b. Can you see you get one here and one here? This, you'd see the dominant trait because the big B is there. That's the general. We'll talk about other cases where that's not necessarily the case. Sometimes you get a combination of the traits. That's either codominance, if they're both of them together, or incomplete dominance, if they have a combination of the two. And that's something we're going to talk about when we get to heredity. But the reason why I talk about this is each one of these alleles has a gene sequence related to it. And that gene sequence is going to be important for making proteins. So the type of protein you have or not have is affected by the alleles that you have. And that's going to be what ends up determining the traits that you have. So the central dogma for DNA basically goes and explains how DNA encodes for proteins. In cells, the genetic information flows from DNA to RNA to protein. Now, as you can see, DNA is in the nucleus. It never leaves. So, if we want to make proteins that we know ribosomes make proteins, right? We have to figure out how does a DNA get its information from inside their home in the nucleus out into the cytoplasm, which is like your neighborhood, right? And it uses messengers. And that makes sense because if you, for instance, if I'm at my house and I want to get something to somebody else and I can't leave my house, I might hire a messaging service. Or a messenger, a bike messenger, for instance, if I need to get a package somewhere. And they will come to my house and they will take it to wherever I need it to go. And that's what this RNA is. It's a messenger. This RNA can leave the nucleus. It won't do it though, unless it's mature. So we'll make it, and then we'll have to kind of change it a little bit to make sure it'll be safe to go outside. It's kind of very similar to what your parents probably did. Yeah, they made you, you were a baby, you grew, you were a toddler, you were a child, and then you became, a young teenager or pre-teen, and then you were a teen, right? And they wouldn't just let you go out into the world. Like, you're not 6 years old wanting to go to see a movie, and they just said, okay, and opened the door and said, good luck, get into the movie. You weren't mature enough to do that. They didn't give you skills and help basically give you everything that you needed to be prepared and safe to go outside. They had to wait until you were mature enough to do it. The cell does that for the RNA as well. Now, the process from going to DNA to RNA is what's known as transcription. And what I think about this is if you ever go into, you've had a traffic ticket, you have anything, and you go into court, there's going to be somebody sitting in the corner with a little typewriter. And they're going to be what's known as transcribing the case, which means they're listening to what everybody's saying in the case because they have to have a legal recording of it. And so they're typing it up. They're transcribing it. Now, if the case is in the US, they're doing it in English. If it's in Mexico or a lot of South American countries, they're doing it in Spanish, right? They're doing it in Italian if they're in Italy. They're doing it in French if they're in France. If you get my meaning, they're doing it in the same language. Now, if you remember, DNA and RNA, these are both nucleic acids. They're the same type of molecule. It's like they're in the same language. Now, the thing is, if you say, for instance, for the most published book in history right now is the Bible, right? And in that, is it in just one language? No. And so what is the process they call when you take a book and you want to change it to different languages, so people that cannot necessarily say, if it's the English, it was originally in Hebrew, right? And so when it was originally in Hebrew and they wanted people who spoke English to read it, what was that called? They translated it, right? If it's in English and it's a book and they want somebody to basically read it in Spanish, or if there's a book in Spanish and they want people to read it in English, right? They translate it. So the process of going from RNA to protein is known as translation. Because they're translating it because proteins, this is a different biomolecule. It's a protein. It's not a nucleic acid. And that happens here in the cytoplasm. So this is what we're going to be talking about, and we're going to start with the first part, which is looking at RNA or DNA to RNA. And you can find that in the next video.